Okay, so if you're looking into a database called ClinVar, where they catalyze, it's an American database where they catalyze all the genetic variants that are associated with some phenotype, like and some disease conditions. So you can see that, of course, the 581 is the most common ones, but there are also other mutations. For example, 656 was catalyzed as another pathogenic variant. There's also other ones that are catalyzed as likely pathogenic variants. And so if we will look into what these genes will express, so one idea to understand that, what the gene does and what the mutations could be doing to the gene is that we want to see where it actually functions. And to know where the gene actually matters, we see what cell types and what tissues that it actually express in. And that's where the gene actually matters to that cell type or the function of that cell types in terms of that tissues. And so here I'm showing you, this is basically a single cell RNA-seq data from the Human Protein Atlas. So basically you're looking at the expression of the ROMBP2s in different cell types from the single cell RNA-seq studies. And so here you can see that, each of the bar is the expression level. So the higher means express higher. And then so on the y-axis is different tissues. And on the axis is different cell types. And of course, it's too small for you to read, so I'm going to try to give a general idea here. So if you're looking at the immune cells, we can see that it is actually expressed higher in the immune cells, and having the dendritic cell being the highest one. And in here is adipocyte tissues, and as well as other tissues. Other immune cells in different tissues is quite ubiquitous in the immune cells in different tissues. And of course, our one target entry cell type is the neurons in the brain. So you can see that it also expressed in the neuron as well as glial cells in the brain. So this is the brain tissues that you can see here, as well as eyes. So it also expressed in the eyes of the neuron and glial cells. And so I think this is our topic, interesting ones, where I think it did suggest it's promising that we can see this gene actually expressed in these cell types. But on the other hand, we can see also expressed in other cell types in other tissues, like epithelial cells in other tissues. So this ubiquitous expression, which means expressed almost throughout our bodies. And could I explain the specific defect that we're seeing in the brain in ANE1 patients? And so it comes down to the very fundamental question is, does ROMBP2 mutation actually cause ANE1, right? Which means we're asking if we carry this mutation, are we definitely getting ANE1? And I think we all know, no, that's not the case. Like there is no full penetrance, which means we have carriers that's not suffering from ANE1. And so even with siblings, like, you with or without this mutation, is some suffering, some doesn't. And so that would be one of the big questions that we want to address is like, why? And so what it suggests is like that mutations that alone is not sufficient to cause ANE1. And then so if itself is not sufficient, and is there other genetic mutation were actually involved that we're being overlooked? So that's where that whole study is going to come in, where basically with your blood samples, what we're going to do is that from your blood, then Natalie's team is going to extract the genomic DNA out from the blood that you kindly given us. Then it's going to send to me, where we will prepare libraries, and there we're going to focus on exons. So exons are basically your genetic sequence that encodes the most important information of the proteins. And then so which is in the red color here. And then so we're going to make a library where that contains the genetic information of all the exons for the 20,000 genes in your whole genomes. And then we're going to sequence specifically just for these regions. And that's going to give us information about 20,000 genes. So right now, we're not just looking at the ROMBP2 one gene alone. Now we're looking at 20,000 genes in your all bodies, and ask like, do we see other mutations that are also associated with ANE1? And then so one interesting idea direction is these genes called GPD genes. And so this is a study published last year And so basically what they're looking at is, so we have the ROMBP2 genes here highlighted in the orange circles. So if you're looking at this genetic locus, it's actually quite interesting. So it's a highly selective region evolutionarily. So here we're looking at, so the top one is the local gene structure around the ROMBP2 in human genomes. So that's where we are. And the study compared that to chimps, which is the species that we're mostly related with, as well as, gorillas, orangutans, and gibbons. And so basically, it's looking at these genetic locus structures across the primates. And interesting, what they found is that, so these regions actually start out by having the ROMBP2 genes, as well as another gene called GCC2. And then it has gone through duplications with these part of these genes. They're duplicating themselves as well as fusion with each other and then expanding. Now that we get a lot of RGPD1, 2, 3, 4, as well 5, 6, 8 in this locus. So it's an expansion. And it is in a way that we don't see, so we see other chimps or the other primate also do that. But these are actually independent events, which means like, for example, the RGPD genes that we have expanded ourselves is actually not the chimp version one. So it's a human-specific ones. And then so what it actually matters is that these are events on the order of millions of years. And so what it matters is like, why it expanded for humans. And in a relatively rapid scale, which usually means that there is a beneficial effect that we're seeing it. That's why we expanded it and contains it, which means like it suggests that it could be functional. Like there is a function with it that's actually favoring bringing increase our fitness of this of our fitness, and so we retain it, expand it in our genomes. And then the interesting part is that, so as I mentioned, these RGPD genes actually was duplicated from the ROMBP2 genes. And so it actually carries part of the N terminals that's actually very similar to the ROMBP2 genes. And that's also where you have these mutations for 5A5 mutations. From the ROMBP1 to 4. Of course, this is from the reference genome that we're looking at, and I believe that we have been looking very deeply for the patients as well as the other close siblings. So one of the ideas is that when I look into the expression of RGPD1s, so for these genes that we haven't known their functions. So we do know that they're expressed, but however, the function is still unknown. But interesting is when we look into the expression of it in the cell types, they're actually quite specific for RGPD1. They're quite specific to neuron and glia cells in the brain as well as in the eyes. And same for RGPD2, where let's see, it goes some neuron, yeah, and glia cells, and again, there's also immune cells, and the blue one is the germ cells. Okay, so basically, taking this together, that what I would like to propose to do is that we want to analyze not only the ROMBP2 mutations, but we want to looking at together with other mutations such as RGPD1s. And then so I believe that would be critical because if you're looking back to Derek's paper, when he first mapped the mutations, that region is so similar that by whole exome sequencing, you actually will have a problem telling, whether you are getting the mutations from the RAMBP2 or from the RGPD genes. However, he does follow up with doing a qPCR where they confirm that the mutation does come from the ROMBP2. But I think the question is like when you have the ROMBP2 mutations, and do you also have that mutations on your RGPDs? Like, I think maybe we should looking at a broader picture of these whole locus. It's not just like one gene alone, which we know is not sufficient at this point. So, and as well as the data set will also allow us to move beyond the RAMBP2. We do have sporadic ones, But I think the sample here is we try to favor the RAMBP2, but then if we have samples that are not RAMBP2 mutations, then we can look into, what are the potential other mutation that's associated with ANE1. So that's what the whole exome sequencing will allow us to do, that we're not just limited by looking at the RAMBP2 genes. And then another thing I propose is that, as I mentioned, there is also limitation for whole exome sequencing. So one thing is that maybe, we can't just look into what happens in the RAMBP2 genes, but also the surrounding, like the RGBD and the structure of that. So maybe we should do something like a more long-read sequencing where we can actually tell where the mutation is actually coming from. And then also there might be, missing deletions or duplication events that actually could be critical, that alongside with these RAMBP2 gene mutations, that I believe the RAMBP2 gene mutations is functional, but right now we know is insufficient. And I think maybe it's because that, you actually have a local specific events that might be occurring in different individuals. And that could probably explain why they were seeing, this not fully penetrate phenotypes. And so with that, I'll end here. Thank you.
Thank you very much. This is super interesting. I have questions as well, but maybe first.
Excellent presentation and different thought processes, isn't it? Because we're very stuck with conditions. But my question is, what does ANE1 deficiency causing human disease? Do we know the spectrum of other conditions?
Yeah, actually, we know quite a little about it. So that's one of the major limitations. I think the study actually heavily focusing on RAMBP2 in this focus.
Patients with ANE1 mutations, is there a human disease?
Oh, so
Okay.
is there a study on it?
Is there something that you might predict from the domain, or is this just because of it's in the same cluster, but is there something like information, the domains look like, whatever the function might be predicted from?
Yes, I think if we do have sufficient data, I think ultimately we will be able to. So right now is
Say the suggested protein of one, whatever. Is there something like a protein domain structure that would already predict, it's functioning in that way?
I think so. Actually, I think people do know to some extent. So it is fused, but there is a big part of it is truncated from RAMBP2. So for example, I think when they're looking at RGPD1, I think it does share the N-terminal domains, but it doesn't have, for example, like E3 ligase domain and the other domains. So I think the other half is more like the GCC, so GCC2, which is the other genes that they seem to be duplicate and fused together and to make other RGPDs. And so one idea is that, how we always see that mutations in RAMBP2, if these genes are also expressed in neuron and glia cells, and then would they be competing or working together, I don't know, with the RAMBP2 itself functionally? And so if they do carry mutations, what could that be affecting it? So I think, and genetically, with a very more repetitive regions, it's always troublesome when you try to replicate, and especially with having them similar, you have a higher chance of recombining, and the recombinations is not, in this case, it's not favoring because, as you recombine with these regions, you might be looking at deletions or, deletions of here and there that's not, that's not ideal.
We actually had a question kind of about this from one of the families. So this particular family, when they were initially tested, they were only tested specifically for the RAMBP2 gene. Then the child had recurrences. So the clinicians pursued whole exome and they found
a variant in RAMBP2. So would we assume then that that was on one of the RGPDs? Or
So it could, so for whole exome sequencing, it's called short read sequencing, which means each of the reads, so each reads you get about 100 to 300 base pair of the sequence of your genomes. And so you're relying on these sequences are specific enough that you can specifically locate it in your genomes. And so when you have genes that sharing the same sequence, then it's going to be troublesome of telling A from B location, you know. So in this case, they are so similar, so it could be the case where they could be from RAMBP2 or from RGPD. And so that would be a question that has to be addressed. But you remind me that another thing is that for patients that already done whole exome sequencing, it could be worthwhile to go back to it and looking at this locus in general. They do have data for it, and to look into to see that, you know, whether you can actually try to map the genetics around this locus, not just for that alone.
so as the support group, if we have families that, which we quite often do, haven't had genetic testing and they only learn that genetic once joining the group, should we suggest that they ask for whole exome testing, preferably?
So at this point, I think I would say that for research purpose, of course, that would be helpful, but for financial costs, then I can't guarantee that I can definitely find the positive findings, but as you can see, these are rare events. So if we can get more and more data, then this definitely would help the research, you know, to actually identify the actual causal, or, you know, in this case, maybe it's not just one causal mutation, but multiple of it.
I have a quick question.
Yes.
So we know that the region where RAMBP2 is located is like a hotspot for recombination, and that's been shown with the duplication with RGPD. So can you comment on cases where there's de novo RAMBP2 mutations in families? Do you think, you know, the fact that the mutation is present in RGPD1 to 4, do you think the de novo acquisition of the mutation could occur through the combination with RGPD?
Yeah, I think that's what what we'd be thinking. Yeah, one hypothesis is that, yeah, like it could, because with the similar sequence, that one idea is that, yeah, when you're replicating or when you're repairing the DNAs, it takes the wrong template. So I think it could happen from there. And given it's so specific to that, then so I think one thing is, you know, would be really helpful is that we're looking across these locus, then will help us to learn whether, you know, for the ones with the episode, what do they look like? And is there a difference from the siblings or the carrying the mutations, but, you know, what the RGPD looks like? And then if there is also, for the ANEmone studies, they also found there's like microdeletions, you know, with a combination event in this locus. And so I believe these will be the things that worth looking into it, that which might be missed previously.
Thanks very much, very interesting. I have a technical question related to long read sequencing. Recently, many of my colleagues have said, well, if you don't have the answer, how about you do some long read sequencing, or you can send it to my lab and I'll do it and it'll take $500 or whatever. So my question is really, is long read sequencing something that we can obtain? We have a number of patients who, not just with RAMBP2 or querying RAMBP2, who have gotten exome sequencing, whole exome, and have not, and it's true for the mitochondrial genetics as well. What's the avenue to that? And this is something that's going to be available clinically.
Yes, I think the major provider that's pushing it is Neoport, which they claim they can do up to megabases. So this locus in this locus alone is 7 megabases. So, and then it is a very complicated genetic, a lot of genetic event happening. So I believe it will actually be very heterogeneous, not just having that mutations with or without it. I think it will actually be pretty heterogeneous in that locations as well. Yeah, so I think in this case, then the long reads is the only way to tell, where you are in this locus and mapping this structure. So I would say, but narrow core sequencing is not the only one. So there are other, Illumina also providing other long read sequencing. I think PacBio is another platform. So either you can go with PacBios. I think they do have settings for more clinical samples. But personally, I don't have experience with that. I have more experience with narrow pore sequencing. So narrow pore sequencing, the way it works right now is like they send you almost like a, like this size or smaller, kind of like a drive, and you just load your sequence. And then they made it in the way that you can do in the field research. So it should be pretty hands-on and easy for that sense. But I do want to say that, yeah, I think in terms of cost-wise, they try to bring it down. So I think in this case, it does make sense, but not in the sense of, you don't know why anything they go for it. It's more like, do we actually need the wrong resequencing? And I think in this case, it actually could matters. Of course, I don't have data right now to say that, but that would be I hypothesize on, and then I think would be really critical if we can actually map the whole region.
And so if you were to suggest a sequence for clinical evaluation, an algorithm, would you suggest first specific gene testing with RAND2 gene, and then moving to the whole exome, and if exome is negative going to long read? Is that the I'm just thinking in practical terms.
Right. So yeah, so ideally, in my idea, so of course, I think ideally what we want to learn is actually, what with our research group, can we then establish what we can find as much as with it? Then maybe we can come up with the AI algorithm to predict it that can actually help us bypass this gene sequencing. Like, I don't think you necessarily need to go with, single whole exome sequencing and long reads. I think by end of the day is that can we actually map to what's the actual critical mutations, right? And then can we have a way to just go right into it, try to minimize the cost? But of course, during this kind of like explanation preliminary phase, we can't do that. But as we learn more from our patients and from our, siblings, like, try to compare them, then maybe that would provide, if we do see, so the question is whether these are linked events with around BB2 mutations or they're actually not linked, right? And then with maybe another thing is that with the de novo, with the de novo mutations, does it occur independently, or actually we're also seeing a link with other RGPDs alongside. And so if we do have a data that can establish that, then maybe we don't have to go through this route.
Thank you, that's very helpful.